Hello everybody, welcome to my channel. This is a pathology video on gastrointestinal pathology. The topic is classification and pathogenesis of inflammatory bowel disease. This is by Dr. C.S.B.R. Prasad, Professor of Pathology, Sri Devaraja's Medical College.
Hello students, we are going to deal with inflammatory bowel disease. This is a chronic inflammatory condition affecting the intestines. And before going into the actual topic, we should know certain fundamentals, that is the oral tolerance, which forms the basis for understanding the pathology or pathogenesis in inflammatory bowel disease. So whenever there is entry of a pathogen or a protein into our system, we react by means of producing antibodies or activated lymphocytes, and this causes inflammation and neutralization of the protein or the organism. There are some sites in the body where bacteria normally exist without any induction of inflammatory process. So these bacteria are called as commensals, which are present in the skin, oropharynx, large intestine. So if we react towards these normal commensals in with the process of inflammation, then our reactions are considered as inappropriate. So why we don't react towards the commensals? So there are proposed mechanisms, and this is due to removal or elimination of the reactive T cells that are produced against this bacteria. And also we produce T cells which suppresses gut inflammation, and this body also secretes what is known as interleukin 10 and transforming growth factor beta, which also helps in tolerating these organisms without causing any inflammation. So in an IBD, oral tolerance is altered, resulting in inflammation. So by definition, inflammatory bowel disease is an immune-mediated chronic inflammatory condition affecting the intestine, and there are two types. One is ulcerative colitis, the other one is Crohn's disease. There are certain characteristics by which we can differentiate ulcerative colitis and Crohn's disease as distinct entities, but in some of the cases the features are overlapping, and it is very difficult to say whether the patient has got ulcerative colitis or Crohn's disease. So such cases are labeled as indeterminate colitis. It forms around 15% of the IBDs. So if you look at the etiology or epidemiology of IBD, incidence, age of onset is one and the same for ulcerative colitis and Crohn's disease, but look at the ethnicity. So Asians are least susceptible for inflammatory bowel disease, whereas other population may have higher incidence of inflammatory bowel disease. Another important thing you should notice is the association with the smoking. So smoking may prevent the ulcerative colitis, so is appendectomy. Appendectomy also offers protection from the development of ulcerative colitis. So if there is anything that is beneficial from smoking, this is the only condition which I can enumerate, but otherwise the smoking is detrimental to our health and it causes early death. So if you look at the pathogenesis of inflammatory bowel disease, there are it's a multifactorial, like say genetic factors are implicated, and epithelial defects and microbiota and immune responses are the ones which are implicated in the pathogenesis of inflammatory bowel disease. But what you should know is, to start with, this diagram represents the components which we enumerated in pathogenesis. So understand the structure of epithelia is very important component in this pathogenesis. So if you look at epithelial cells lining the intestines, they are all bound together by means of tight junctions, and because of the presence of these tight junctions, this epithelia is impervious to the elements derived from the bacteria, and anything that alters this tight junctions may be associated with increased permeability of the intestinal barrier. This is the basis for the development of inflammatory bowel disease. And once this permeability is increased, the bacterial components may enter into the subepithelial region, and this again initiates inflammatory reaction wherein macrophage, T cells, B cells will come into picture and produce varieties of cytokines which again may affect the bacteria, which again may affect the overlying epithelium, causing more permeability. So there is defect in permeability to start with because of tight junctions are affected due to any reason, and because of this increased permeability, bacterial components gain entry into the subepithelial region, and this immune cells will react to this bacterial component, producing varieties of cytokines which again go and damage the overlying epithelium, thereby causing more and more increased permeability. So this is the basic understanding of pathogenesis of the inflammatory bowel disease. How this permeability is altered and why the bacterial components which are considered as normal commensals are considered as foreign and activating the macrophages and immune system. We will be dealing with this later. So pathogenesis again, if you look at the components, genetic factors, epithelial defects, microbiota and immune responses. So coming to the genetic factors, IBD is a polygenic disorder and more than 160 genes are have been identified and which are associated to be associated with IBDs, and 1/3 of them belonging to ulcerative colitis and Crohn's disease overlap, and because of this overlapping, probably they have sometimes the common features which makes it very difficult to categorize whether the patient belongs to Crohn's disease or ulcerative colitis. And look at this genetic association with other immune mediated disorders. So IBD is most commonly seen in those persons who are suffering from rheumatoid arthritis, SLE, psoriasis, ankylosing spondylitis and type 1 diabetes mellitus. So the autoimmune disorder may be the basis for the development of inflammatory bowel disease. So several of these IBD associated genes overlap with the genes involved in our response to Mycobacterium tuberculosis and Mycobacterium leprae. If you recollect, the type of inflammation that is seen in Mycobacteria induced infections, they are granulomatous reactions. So because of this, probably Crohn's disease responds mainly in the form of production of granulomatous inflammation. And this is an important slide wherein NOD2 gene polymorphism is strongly implicated in the development of inflammatory bowel disease. So this NOD2 gene is responsible for integrity of intercellular tight junctions, and also it produces a protein, intracellular protein, which binds with the bacterial peptidoglycan that stimulates the signaling pathways. So any defect and polymorphism in NOD2 gene may result in defective tight junctions and defective response to the bacterial proteins. So other genes that are associated specifically with Crohn's is ATG16L1 and IRGM, and both are autophagy associated pathway dictating genes, and probably thereby causing abnormalities in the way our cells deal with the bacterial organisms. And again, these are some of the genes, chromosome location on the chromosome, and also their association, whether it is predominantly Crohn's disease or ulcerative colitis or combined. Coming to the second component, epithelial defect, I told you already the epithelial defects mainly involves the defective tight junctions. So again, NOD2 polymorphism is responsible for this, and the ECM1 polymorphism is linked with specifically ulcerative colitis. This extracellular matrix 1 inhibits in a normal person matrix metalloprotease 9. So any abnormality with extracellular matrix protein 1 may not inhibit matrix metalloprotease 9, and hence the damage will occur to the epithelium. And another gene is HNFA polymorphism, which is again linked to ulcerative colitis. And interesting point regarding this HNFA is its association with MODY, that is maturity onset diabetes in the young. So this is again this in this disease the patient also shows, in addition to the diabetes, defective intestinal barrier. So probably this is an interesting point with respect to this HNFA. And microbiota, not to polymorphism. I told you not to is associated with the production of certain proteins which interact with the bacteria and stimulate the downstream cellular responses. So not to polymorphism may exaggerate this type of response, and mainly it is seen to be associated with Crohn's disease. And another important point to say that microbiota is implicated in this pathogenesis is the presence of antibodies against flagellin. So presence of flagellin antibodies is considered to be one of the markers for Crohn's disease. And interestingly, if you transplant normal microbiota from a normal healthy individual into the IBD patient, and their symptoms get ameliorated, and probably they may improve in their condition. So coming to the unified theory, so combining the genetic factors, the microbiota and epithelial integrity and immune responses into one theory. So this theory proposes there is a epithelial barrier dysfunction wherein which increases the permeability of the epithelial barrier. Probably genetic factors play a role in this increased permeability. Once there is increased permeability, bacterial components enter into the subepithelial region, and that immune response cells react in an abnormal fashion, producing cytokines, which again goes and attacks the overlying epithelium, further increasing the permeability. And ultimately, there is this microbiota also play a role in production of this inflammatory bowel disease. So again, the same diagram. So the epithelial membrane integrity is very important, and this is maintained by cellular tight junctions, which are affected by the genetic mutation, and thereby increasing the permeability, and further course of action will follow. And here, interesting to note that the macrophage T cell cooperation results in activation of T cell pathways, which ultimately produce interferon gamma and tumor necrosis factor, and also Th2 differentiation may be associated with interleukin 13 production. All cause the damage to the overlying epithelium. And there is a production of interleukin like interleukin 13, which stimulates, interleukin 23, which stimulates Th17 cells, which are further associated with the secretion of interleukin 17, recruitment of neutrophils and activation of neutrophils, and damage to the overlying epithelium will be facilitated by this process. So this slide tells that genetic association of inflammatory bowel disease with the autoimmune disorders, mainly psoriasis, ankylosing spondylitis, and primary sclerosing cholangitis.
Thanks for watching. Please subscribe by clicking the button subscribe.